Okay, I think we'll make a start now. Welcome everybody to this event hosted by the National Center for Research Methods. My name is Tarani Chandola, and this is an event on what can social scientists learn from the omics revolution. And I'm very happy that we have three very great speakers here today, Professors James Nazroo, Professor Melinda Mills, and we'll shortly be joined by Professor Gillian Bentley. Before I introduce James, I just want to give a few background notes on why we're having this event today. That's partly because of the omics revolution, which is relates to sort of genomics and metabolomics, proteomics, all these sort of new biological measures that are now being incorporated more into social science data sets. And social scientists now are sort of learning more about how they can use these kinds of data. And I originally conceived of this event as both in terms of how key social scientists have learned from being able to use these kinds of data and also contribute to the debates around their use. And welcome particularly to those people who are joining online. If you have any issues with the live stream feed, please let us know through the Twitter account for NCRM, so that's at NCRM UK, or let us know any questions that you might have through the NCRM Twitter feed. So first of all, I'd like to invite Professor James Nazroo from the University of Manchester to talk about how interdisciplinary research and omics data can work together. Thank you.
Thank you, Tarani, and thank you to the NCRM for inviting me to do this, or to contribute to this session on, I guess, sociology, social sciences, and the omics revolution, and how we as social scientists might make use of omics data. Just a little bit of introductory context. As you can see, I'm going to talk about interdisciplinary research, or use the notion of interdisciplinary research to frame some of the things that I want to say. In terms of omics data, I'm going to focus particularly on metabolomics data. But as you will have gathered, I am not a biologist. I'm a social scientist, a sociologist, and even though I have had some training in biology, I come at this from a relatively naive perspective. Which means that what I tell you about metabolomics will be very basic, but hopefully enough to give you a sense of the value of those kinds of data. So a non-expert introduction to metabolomics data from a social science perspective, and particularly from a kind of critical social science, how might we make use of such data, and what value is social science in this context. So a brief overview of what I'm going to do. So the context of my talk is a large interdisciplinary program of research called the FRAIL study, which stands for Frailty, Resilience and Inequalities in Later Life. This is a study based at the University of Manchester, funded under the Lifelong Health and Wellbeing Programme, led by several of the research councils that fund research in the UK. And it involved a big interdisciplinary team I'll introduce at the end of my talk rather than at the beginning, but covering a number of disciplinary perspectives and making use of a range of omics data. And so it provides a very useful context to begin to think about this set of topics. I'll say something about biology and social science research, but really about the biological pathway that we're focusing on and how that fits into social science research. I'm going to say something about social inequalities and why social inequalities are crucially important, and why we need to understand them from a social science perspective, but where biological pathways might be useful in shedding light on those. I'll give a bit more of a detailed account of metabolomics and how at least I understand metabolomics. I'll make some errors and I'll read a bit when I give my account of that. I can't do it off the top of my head, but hopefully it'll be enough for those people who are not familiar with metabolomics to get a sense of the potential of those forms of data and how they might be generated. I'll say something about interdisciplinary working and how we might approach interdisciplinary working. And before I start, I'm just going to say something about the orientation of my talk. And so the orientation of my talk is around social science problems. and trying to understand the nature of social science problems, causal processes related to social science problems, and then how we might think about interventions for social science problems. Here, of course, I'm particularly interested in the question of inequality. So inequalities, and the Frail study, inequalities in later life. So how we might understand inequalities in later life, what drives inequalities in later life. That's the focus. And then where does biology fit into that process of understanding? So this is really thinking about biology as a part of a complex pathway, and how biology fits into our understanding of complex social pathways, social causal pathways. My talk is not really focused on the ways in which biology might explain social problems. And there is an important distinction there. So I'm not making a claim for biology being the solution to understanding social problems. In fact, I would make a claim for the opposite. But rather that by understanding biological pathways, we may elucidate better social and economic pathways. So that's the framing of my talk. So I'll start off by introducing the Frail study. So this is our research agenda as we set it out at the beginning of the program. So as I've kind of already introduced, our focus is on inequalities in later life. We take two broad concepts, two broad outcomes, frailty and well-being, to model inequalities in later life and to try and understand the social and economic drivers of those inequalities in later life. Important point to make, and it's an important point that I'll return to at some point, is that in order to do this kind of work, you need very good measures of the outcomes that you're interested in, of the phenotypes you're interested in. So a large part of our program is about theorizing and measuring the concepts of well-being and frailty. Thinking about a range of contributions, a range of factors that might contribute to well-being and frailty, and consequently to inequalities in such outcomes. So this is where biological pathways begin to fit in, but also where a whole range of social exposures and the ways in which they trigger pathways is important to consider. Interdisciplinary approach, and we go from genetic to metabolic to psychological to social and economic. I haven't put economic on there, but economic as well. A life course approach, thinking about later life, of course, within a life course context, but not just early life, also thinking about transitions in later life, how they might be shaped by early life, but also how they might be shaped by things that happen in later life as well, and trajectories across the life course. And then it has a comparative dimension to it, which again, for a lot of the kind of research that we do using omics, is very important to be able to reproduce the things that you find in other studies. And here we have a couple of other studies across the world that are very similar to the data source that we use for this study. Which brings me to the data source, of course. And so the data source we use is the English Longitudinal Study of Aging, a general population panel study of people aged 50 plus. A brilliant study for this kind of integration of social, biological, and omics and non-omics research. I would say that. I'm one of the investigators of ELSA, so I would say it's brilliant. It is actually a really very, very useful study for multidisciplinary and interdisciplinary working. So I've given an indication on the slide of the kind of topics that we cover: demographics, economics, measuring health, cognitive function, mental health and well-being, and social participation, social networks, and so on. What's really valuable about ELSA is that alongside these measures, we also then have biomedical measures. So not as frequently as the interview, we have a nurse visit. And at the nurse visit, there's a measurement and measurement of the participant and collection of biomedical samples from the participant. And two very important things in the context of the study that I'm going to talk about is that it's including DNA collection, and it includes the storage of serum and hair samples, which can then be analysed further. We have a life history approach, which enables us to do some life course work, and I've already mentioned the comparative potential there. And as I've said already, the intention is to develop an understanding of complex pathways that are studied separately by different disciplines within a social and economic context. This gives a model that was drawn by one of my colleagues. This model was drawn by one of my colleagues and gives an idea of the biological pathway that we're interested in. So we're interested in the importance of the genome, how that relates to molecules, how that relates to the biology that we can directly measure, plasma molecules and the metabolome, and then how they relate to the phenotype that we're interested in. And here we've got frailty and well-being as the key measures, but also there's a whole set of other biological measures that you could be potentially collecting, and there are, of course, some questionnaire measures that you can study as well in these kinds of studies. But it's the top half of this, so the genome, plasma molecules, and metabolome that I want to focus on in a minute. At one level, we did this in a theoretical way. So we started this study before we knew that we would have GWAS data available. And so we did this in a theoretical way, hypothesized pathways around the HPA axis, so around cortisol and so on, and around lipid metabolism, were the hypothesized pathways that we intended to pursue. And so we had a focus on candidate genes, we had a focus on particular hormonal assays. But with the implementation of GWAS within ELSA, then we had the opportunity to do work that wasn't driven by a priori hypotheses. And that's where the metabolome became particularly important in our study, as I'll go on to show you. Just to say something, and now I'm going to have to start reading a bit because I don't know all of this off the top of my head, just to say something about the kind of biological pathway that we're interested in investigating. And again, this is a slide that I've taken from one of my colleagues. And so we start off with genetics, and I guess many of the audience will be familiar with the use of genetics to some extent within social science studies. It's becoming increasingly popular, of course. And genetics we can think about in terms of the candidate gene association study that I very briefly introduced, where you have a hypothesis around particular genes that you want to measure because you think they relate to the pathway that is of interest for the phenotype that you're interested in. So you may have a hypothesis about what causes frailty and the biological mechanisms involved, and therefore which genes to study. Or you may want to do a GWAS study, so there you have no clear hypotheses, you're exploring as much of the genome as you possibly can and looking for associations with the outcomes of interest. You can go further and do genomic complex trait analyses, where you take the whole of the genome that you've measured and see how much of the variance it explains in the outcome that you're interested in. And you can do the opposite, do it the other way around, measuring genes within individuals, knowing how those genes contribute to the outcome, then give the individual a score that assesses, that summarizes their genetic risk for the outcome that you're interested in. Epigenetics, the ways in which the genome becomes modified through a set of biological, perhaps environmental influence processes, and therefore bits of the gene operate in different ways as a consequence of that, and that varies across individuals, so trying to understand that variation across individuals. Transcriptomics, which focuses on RNA and the ways in which genes implement RNA activity, the things that make a difference, and then produce proteins. Proteomics, which is kind of large-scale assessment of the proteins that are in the biological sample, be that blood or whatever, and how that product of the gene, so this is the product of the gene activity, and how that product of the gene relates to your phenotype. And then metabolomics. And metabolomics, I'll just say perhaps slightly more at this stage, and I'll say more later in terms of metabolomics. So metabolomics is in a sense a study, a systematic study of the chemical composition of the biological sample that you're interested in. So it could be a cell, or it could be blood, or it could be a tissue sample. So it's a consequence of cellular processes. It's what's left behind as a result of all of the genetic and biological activity that's happening within cells. And in that sense, it reflects two things. To a certain extent, it reflects a phenotype. This is what you're looking at. This is the end point of the biological processes. But it also measures process in time. So it's measuring both processes leading to the outcome and the outcomes itself. And it's important to bear that in mind when you think about what I'm going to say as I continue on in my talk. So it is an outcome in itself, but it's also a set of processes that, or reflects a set of processes that relate to other outcomes that you might be interested in. So sometimes I think of it as the distal phenotype, and sometimes I think of it as a distal part of the biological pathway. And I guess for us using these forms of data, not just the metabolomics, for us using these forms of data, an important question then is how do we integrate these different levels of measurement in terms of trying to understand the phenotypes that we're interested in. So how do we integrate genetics, epigenetics, transcriptomics, proteomics, and metabolomics, and which points of these processes do we actually want to assess and why in terms of trying to understand a biological pathway? That's a kind of throwaway comment a bit. I won't say very much more about that, but I can return to it perhaps later. Okay. Just want to say something about inequalities now and why we're studying inequalities, and to kind of calibrate the biological and the contribution of the biological to the need to understand inequalities. And I'm going to show a set of very, very simple outcomes, but just to make the point about inequalities. So here we have a measure of, so this is a well-being, age and wealth chart. So it shows well-being across ages. It actually is longitudinal or semi-longitudinal analyses. Well-being across ages, stratified by wealth. So the top line are the poorest fifth of the population, and the bottom line are the richest fifth of the population. What I'm measuring here is actually the converse of well-being, or one converse of well-being, depression, or depression symptomatology. And what you can see is the U-shaped relationship, which I think has been classically presented now for a number of years by a range of researchers. The most important point from the issue of inequalities is that although there's this U-shaped relationship, the most affluent have a much lower level of depressive symptomatology than the richest across the age spectrum, but even at the lowest point of depressive symptomatology for the, oops, I was trying to find a pointer. I have a pointer. So even at the lowest point of well-being for the least affluent fifth, there's no real overlap with the levels of well-being for the most affluent at their highest point of depressive symptomatology. Two distinct populations in terms of their experiences of depressive symptomatology. This looks at frailty, which is the outcome that I'm going to talk about most when I show you some empirical examples of metabolomic work. So these are frailty trajectories by cohort and wealth. It's essentially the same chart as the one you've seen before, or the same idea of a chart as the one you've seen before, except here I explicitly show cohort differences as well. What's important is that the levels of frailty for the most affluent, and this is the most affluent third, not the most affluent fifth, are so much lower than the levels of frailty for the least affluent. And the horizontal line there gives you some sense of the difference in ages at which a particular level of frailty is hit. And you can see that it's a kind of 10 or more than 10-year difference. There are other important subtleties here in this chart, but that's the key one. Again, a marked difference, an important social problem that we should understand. And this kind of emphasizes the point a little bit. And so this shows your risk of becoming frail, but frail is not an end point necessarily. People recover from frailty, and particularly if you use a dichotomous measure of frailty, you can see that recovery happening. People move forward and backwards across the border of frailty. And the top table up there, which is unstratified by wealth, shows that for the people who are not frail at a particular wave, 15% of them become frail at the following wave, which in ELSA is 2 years later. But the frail, the bottom line, and the blue number, shows that 16% of people recover from frailty in 2 years, in a 2-year period. Then if you look at the least affluent, so the bottom left table, you can see that 26% of the not frail become frail, compared with, on the right-hand side, 10% of the most affluent. And then amongst those who are frail, there's a recovery rate of 10% in the least affluent, compared to a recovery rate of 28% for the most affluent. These are descriptive findings, but they show very marked differences in risk of frailty, and risk of onset of frailty, and differences in recovery from frailty for the most and least affluent. Okay, so by way of introduction, that is, and I'll say more about the social science problem as I conclude my talk. Now to talk about biology in a little bit more detail, and given time, I guess you'll be relieved that I'm finally getting to biology. So biological pathways to frailty, and I'm first going to show you the problem that we have. And you may not think this is a problem, but this is the problem that we have. So what we have here are particular plasma molecules that we were interested in terms of our hypotheses around lipids and frailty, and our hypotheses around inflammation, HPA axis, and frailty. And so the top half of the chart shows that for 3 of the outcomes we were interested in, cholesterol, HDL, lipids, and LDL lipids, we see a decline in the presence of those on average as people become more frail. So towards the right of the chart, people are becoming more frail. But triglycerides rise a little bit as people become more frail, suggesting important biological pathways are going on here. But actually we don't know what the nature of those pathways are, because what we're doing is looking at a semi-distal outcome. And the same with the chart below, which looks at some inflammatory markers and a few other things. And so what this shows is that for fibrinogen and for white blood cell counts, we see a rise with increasing frailty. And for ferritin and for DHEAS, which is a hormonal component, we see a decline in levels with frailty. Again, we see there are important biological processes happening here, both in terms of lipids and in terms of inflammation processes. And they're processes that we find in the literature as well, but we actually don't know what the mechanisms are that are driving these. And we can show social correlates with these, socioeconomic correlates with these, but again, we don't know what the mechanisms, what the underlying biological mechanisms are. And so the approach of metabolomics is one way of beginning to get to some of the biological mechanisms there. And so that's what I want to talk through now. And again, I'm going to have to refer to notes as I go through this. So what do we do when we do metabolomic methods? So I think I've already said, or perhaps half said, the metabolome refers to the set of small molecule metabolites that are found within a biological sample. These are the products of metabolism. They might be partway through the process of metabolism, or they might be the end product of metabolism. I consider them, I think most people who do metabolomics work, I don't do metabolomics work, but most people who do metabolomics work consider them as downstream markers. So these are distal markers reflecting the consequences of intracellular mechanisms, so the products of the processes started by genetics operating within the cell, but obviously mediated or moderated by environmental pathways, triggered, moderated by environmental pathways, not mediated, triggered, moderated by environmental pathways. And the argument might be that if we can identify these metabolites within a biological sample, then we can identify biological causal pathways. And we can also potentially, if we think of these as a distal process, we can also identify biomarkers of the outcome that we're interested in. If there's some difficulty in measuring the outcome we're interested in, then these provide robust, repeatable, objective, quantifiable markers of those processes. If we're convinced they're good markers of those processes, of course. And the argument is that every cell has a particular metabolic fingerprint that can be elucidated, and it can be done on organ or tissue-specific information, or it can be done studying biofluids, which gives a more generalized indicator of the biological processes that are operating within the biological sample, the individual that we're studying. And my colleagues who do metabolomics in a whole range of types of biological material. So some study exhaled breath, some study urine, some study blood, and some study tissue-specific samples, and so on. Important point to note is that we take these samples at a particular point in time, a particular second or set of seconds in time, and so they reflect a biological process at that particular point in time, rather than giving a more holistic across-time process. So repeat measurement is a very valuable thing to do, and we have done some repeat measurement in our work. In order to do it, you get the biological sample, you process it, you store it, then you process it again, then you retrieve it, you process it again, and then to get to measure the metabolites, you do gas and/or high-performance liquid chromatography. You then do mass spectrometry to identify the items, the things that are measured as a result of the chromatography, and you can do that either by mass spectrometry or more contemporaneously, people are beginning to use nuclear magnetic resonance spectrometry. You can see these are words I'm not very familiar with using. One of the things that really struck me, I'm not used to biological or lab work, as you can imagine. One of the things that really struck me was the effort that my colleagues put into the instrumentation that they were using, and making sure that the instruments were performing in reliable ways, as well as valid ways, but reliable ways over time across different samples, and so on. So the standardization and calibration of instruments was a huge effort on the part of my colleagues. Once you get your output, you then can compare it with known values. Known values come from reference data sets, which tell you what you're measuring. Typically, you do this in an untargeted manner, so you don't have a particular interest in you don't have an interest in any particular metabolite, rather you're measuring whatever metabolites that the sample and the instruments allow you to measure. The metabolic composition that you get, typically, if you do all of the different types of chromatography, typically give you thousands of variables. These are thousands of outcomes or thousands of predictors. In order to deal with those thousands, typically data reduction techniques will be used because they correlate with each other. So principal components analysis, they reflect similar biological pathways. So principal components analysis is typically used to identify those connections, to identify patterns. Then you use biological network modeling, so known biological networks known biological processes to understand the relationships between the things that you've measured using the metabolomics process that I've just described. I put the word mummy chog in there, not because I understand what it is or how it's done, but just because this seems to be the so that you can have that as a reference term. This seems to be the thing that most people use when they're doing this, mummy chog network analysis. And then once you've identified potential metabolites and the networks that they're based within, you can then correlate that with the phenotype of interest, frailty. So the work that I'm going to describe to you is untargeted metabolic phenotyping of blood serum that we've obtained from ELSA participants using gas and high-performance liquid chromatography. I'm just going to show you the outputs from the, I think, positive high-performance liquid chromatography, followed by mass spectrometry with a sample of just over 1,000 people who were studied in ELSA. And this is the kind of output you get once you've got through the first phase of the analysis, or once your colleagues who know how to do this have got through the first phase of the analysis. And again, I'm going to have to read a little bit to explain this to myself as well as to you. So this is a principal components analysis. A principal components analysis using principal component discriminant function analysis, which is intended to separate out individuals on the basis of the principal components that are identified in their metabolomic profiles. And here we stratify the individuals according to four levels of frailty. So the blue have very low scores on a frailty index, the green have quite low scores on the frailty index, the pink are moving into a frailty syndrome, and the red are clearly have a frailty phenotype according to the measure of frailty that we use. And as you can see, you find a separation between the first two and between the first two and the second two on this analysis, which uses two components. So there are two components to the discriminate function principal components analysis, and according to those two components, at the lower left segment are those people who have a reasonably high score or a very high score for frailty. The right-hand segment are those with a very low score for frailty, and the upper segment are those with a modest score for frailty, not really frail but have the beginnings of some symptomatology. So this shows, I think, quite clearly that if we do this kind of work, then we can correlate it very well with the outcome of interest. You could think that you might be able to use the outcomes of this work to provide biomarkers for frailty. Once you've done this, you then know what the components that are within discriminant function 2 and discriminant function 1. So you know what the variables are that are driving the principal components that are there, and you can then plug them into a biological network. And I apologize for the nature of this slide. A biologist would understand it, would value it probably. I think we can't. We can only value it to the extent, or at least I can't, we can only value it to the extent that a biologist tells me what's important here, or what the commonalities are here. So we have the two functions, module A, module B, and within those two functions we identify things that are being measured that correlate with those functions, and then we try and identify those that relate to the similar biological pathways. And what we find in there, in both of those functions, is something related to vitamin E. So those items that you might be able to see that are red and green, and they're present in both of those modules. And then those that are related to the carnitine shuttle. Those that are orange, and I think that's grey, it might be pale blue. And they are only in module B. And then this gives us a clue to the biological pathways that are relevant to frailty that we can then kind of theoretically check, are these important? And that's what this, in a sense, next step does. So it's not a next step, it's an attempt to characterize how these might relate to each other. And what this says is, again, referring to my notes, so the mechanisms that are outlined in these two blocks, the top block is vitamin E antioxidation, the bottom block is the carnitine shuttle. What this shows is the proposed mechanisms that demonstrate how the antioxidant properties of vitamin E analogues, the things that were directly measured, could protect fatty acids from liquid peroxidation within the cell. So with more fatty acid available, the carnitine shuttle, the bottom part of this block, has more substrate that can be processed to produce something called acetyl-CoA, which is vital for energy production. So what this says is that within the frail metabolite, there's a lower abundance of vitamin E analogues and carnitines, which then produces a downregulation of the system that you see in front of you, and hence a lower energy output. And you can see the theoretical fit then from a biological level with the notion of frailty. So the intention is that this sheds light on the pathways related to frailty, potentially finds biomarkers for frailty, and elucidates the pathway particularly well once you put it into the broader process that I discussed. So going from genomics through transcriptomics to metabolites. Okay, I'm now going to move on and step off the uncomfortable territory that I've been on, for myself at least, and talk a little bit about interdisciplinary research. And just to say something about context, so I've described a very complicated interdisciplinary program of work that goes from cell biologists through to sociologists, economists involved, social statisticians, clinicians, and of various types and so on. Why do we do interdisciplinary research? And perhaps you might want to pay most attention to the final two bullets here, but there are theoretical reasons for doing it as well, or scientific reasons for doing it as well. And the argument is that if we are interested in something like inequalities and something complex like frailty, then these are complex concepts, complex processes with interlinkages between processes that are typically studied by different disciplines. So that requires an integration of fields of knowledge from across disciplines, and not even multidisciplinary approaches, an attempt to integrate different disciplines' work. So moving beyond traditional scientific boundaries or schools of thought and creating new knowledge, and hopefully more than the sum of parts to try and understand, to try and head towards common research goals. So research goals that have at least some level shared interest from across the disciplines. Thinking outside the box, moving beyond ontological and epistemological boundaries. So thinking differently about cause, thinking differently about methods when framing our questions. It's particularly important to do that when you're framing your questions, and leading to unique ways of understanding what's going on. And if you really take this seriously, then you think about your questions and then you revisit your questions to frame them in ways that reflect a broader set of thinking, placing complexity and that broad understanding at the center of the questions that you're asking. Why do we do it also? We also do it because our funders tell us to do it. Our funders tell us to do it for good reasons, but it is a challenge for us to do it. But if you are given funding to do it, then obviously you have an additional incentive or an additional means to do it. And of course, it's very important in terms of trying to have an impact beyond the academic world, but also maintaining our profile within the academic world. Am I going too long? Risks and challenges, I don't have actually very much more to say, I think, so if I can do it quickly. Risks and challenges in doing this work. So I've already made the argument, if the processes we are investigating are multi-dimensional, then it makes sense to do it in this way. Our approach to understanding has to be multi-dimensional. We have to push ourselves outside of our disciplines. But when you do that, then you are having to reconcile different and often conflicting epistemologies and different ontologies and often conflicting ontologies. Integrating different ways of thinking about data, thinking about knowing, and different ways of thinking, theorizing about explanation. So different ontologies, different worldviews, contrasting ways of understanding what's going on. How do you get the biological understanding together with the clinical understanding, together with the sociological understanding of what the explanatory processes might be? Different epistemologies. Who cares about questionnaire data? Who cares about experimental data? So the kinds of experimental data that a biologist might produce may, from my particular perspective, be entirely decontextual and therefore of limited relevance. Whereas the kind of data that I produce might actually not be robust enough to get to causal processes from the point of view of a biologist. So we have to be aware of these differences, and instead of just disrespecting each other, understand the context within which we're operating. Threat to academic position, all of a sudden, and you've already gathered a little bit of my discomfort, though I have rehearsed, a little bit of my discomfort in moving to a territory that I really know nothing about. If there was someone who knew about metabolomics in the audience, there may well be, of course, they could no doubt have pushed me very badly in relation to that. I become a novice, so I'm an expert in my field, but I become a novice all of a sudden. And not only am I an expert, but not only that, I'm an expert in my field, but all of a sudden someone who's a novice to my field actually asks me something that is really impossible for me to answer because they're informed by a different set of theoretical propositions. And I find that even with economists. So when you think about working with a clinician or a biologist, how that might happen, and you have to think about what you're doing completely differently as a consequence. So there is a real threat. Publications, how do we publish together? What kind of publications would we like to achieve? Which journals? Which are we allowed to do monographs, book chapters? Who are authors, how authorship is decided, and how the hell do you get interdisciplinary papers? Those are really big questions for people doing interdisciplinary work. But, and I come back to the opportunity again, there is an opportunity to do stuff that is different and perhaps more groundbreaking and perhaps more useful if we do interdisciplinary work. How to do it? Team working. I put the word power here because I think power is really important in these teams. Power is important even within a single disciplinary team, but it becomes particularly important in interdisciplinary teams. Most important is communication, learning each other's languages, understanding what you talk about, thinking about the acronyms that are being used and so on. So a methodological and a disciplinary multilingualism is really important in terms of, but it's a learning process. You only get that through regular meetings, regular discussions, trying to understand the outputs of different arms of the research. Different work practices, the way in which you work in a laboratory, the hours you keep, how you do it is quite different from the way you work if you're doing desk research, which is quite different from the way you do it if you're doing interviews. So understanding that people might not be around in the ways that you might expect them to be around, or they might have demands on their time in ways that you might not be expecting. Leadership, respect and collegiality. I've talked about that a little bit in the previous slide, but thinking about this as partnerships rather than a discipline being used to solve the needs of another discipline or support the needs of another discipline. So even though frail is a social science-led study, I don't think we've ever interacted with each other as though the biology is just to help us. There's lots of important biology that's going on there in its own right. And related to that is the need for seniority across the arms of the study. So it's quite often that you bring in a junior from one discipline to help you. That just disempowers that junior person. So ensuring they have the right mentorship, the right support. And back to this respect thing again, theoretical and methodological respect. All right, I think I'm just going to conclude now. I've got 2 slides of conclusion, and one is about measurement and methods. So I've talked a little bit about epistemological issues across disciplines. What we need to remember, of course, is that none of us are methodologically perfect in the work we do. So we have to understand the problems not just in our field of research, but also in the other fields where the other disciplines that we're working with, if we're going to do interdisciplinary research. So if we're using genomics or omics data, if we're using omics data, then we have to understand the set of processes that influence the quality of omics data. So the way the data are collected, the way they're stored, the way they're analyzed, the quality control implementation that's happened in that process of analysis. Sorry, that's the analysis to produce the outputs that are then statistically analyzed. So the set of issues that are related to statistical analysis of big data, and often big data with small effect sizes, so dealing with the number of statistical tests we do, thinking about the ways in which we might understand effect sizes that as a social scientist you would consider to be unimportant, but are not. And also thinking about the ways in which the things that we're really interested in, protein expression in this case, result from a number of different biological processes, a number of genetic effects. On the other hand is the measurement of the phenotype. So making sure that you have high quality measures of phenotype. There's no point having good biology if you have crap phenotypes. So making sure, and I haven't gone into it, but the ways in which we think about and measure frailty, the ways in which we think about and measure well-being in the example that I showed you, but also all the others. And so as I say in the final bullet point here, the phenotypes that we're interested in the social sciences are complex. They have many relevant biological pathways driving them, and a wide range of social and environmental influences, and they vary over time. So we're not really interested in the discrete time point measurement, but really how these things evolve over time. And I've given you a few examples, and if you just think momentarily about any of those examples, depression, hypertension, frailty, weight, fertility, you can see the points that I'm making in terms of the complexity of those phenotypes and the robustness that we need to approach theoretically and empirically, the robustness with which we need to approach them. And then my final slide, a return to the importance of social science and the importance of inequality. So I've made the point there are significant socioeconomic, and I haven't talked about ethnic, gender, area, etc., inequalities that continue into later life. They're relevant for almost all of the outcomes that we might be interested in. Almost anything we might be interested in has these very marked inequalities. The social phenomena that we're studying are complex. So the ways in which they occur and the ways in which they're experiences are complex. So if we think about the things that might be important in later life, that relate to the outcomes that we're in later life, marital status, health and disability, patterns of retirement, employment, social and civic participation, even wealth, are very, very complex concepts, and the ways in which they change over time are very complex. That's the kind of phenotypic argument, that we do have to measure our phenotypes well in the social sciences if we're going to get anywhere. But then the processes, the socioeconomic processes that drive them, are also highly complex. Interrelated processes. They're not a single discrete process. So I've identified some that I think are particularly important in later life: material, economic well-being, social and cultural capital, employment quality and retirement processes, and social status and valued identities. And so if we think about those processes and the ways in which we might understand them, it's clear that biology might help us understand the pathways, but they don't let us understand at a fundamental level the ways in which inequalities operate in later life. So my argument here is to pitch for using biological pathways to help us understand the broader socio-economic fabric of our society and in the ways in which that leads to inequalities. So thank you very much. Oh, sorry, a very important, I did say I would briefly mention, you can see there's a very large list of people who have been involved in this work at various levels, various stages. So they include people who are doing metabolomics, they include people who are doing social statistics, people who are doing psychiatry, people who are doing sociology, people who are doing gerontology, let me see if I can see it now, and indeed people who are doing endocrine work. And there's probably one or two other specialisms in that group of people. Thank you.
Thank you very much, James. We have time for some questions. For those of you who are joining online, you can tweet your questions to @NCRMUK. I'll keep an eye for any questions that are coming on, but for people here, if they've got any questions.
Yeah, so I understand the link between frailty and inequalities, and I understand biomarkers, some of the metabolites mentioned. What particularly are you hoping, what novel insight are you hoping to gain from using biomarkers? Just basically another sign that they are, on a predictive path to develop to be more frail than others. Sort of anything beyond just using another marker, even whether it's a biomarker or the socioeconomic marker. Okay, but is sort of any additional insight that one might hope to gain from using biomarkers or other biosocial data?
Yeah.
Could you repeat
I need to repeat the question. Oh dear. Okay. This will show my ability to pay attention to the questions I'm being asked, or just to extract the bits that I really like. So the question here is the metabolite is a biomarker reflecting quite closely, if you accept the data that I've showed you, the phenotype that I'm interested in here, frailty, and what does that do beyond provide me another marker of frailty in sum? And I guess my answer is twofold. Of course, I focused on the metabolite because other people are talking about other bits of this biological process. But in a sense, what we're trying to do is to get reasoned biological pathways. So evidence for particular biological pathways that at a theoretical level, our understanding of biology networks and the ways in which they operate makes sense in terms of frailty. And so if we can do that, if we can find the genetic relationships, we can find the relationship with the hypothesized markers of the HPA axis and lipid functioning, then we might have a sensible biological pathway here, which then helps us understand the biology of frailty, which is what the biologists, of course, are particularly keen on doing. So we find the genetics through to the measured questionnaire phenotype, a biological pathway. I think the other value is that if we can get good omics markers of the outcome, and here I'm talking about metabolomics, but other good omics markers of the outcome, then something that is a complex clinical phenotype can actually be measured in a much more straightforward, reliable, robust way. So that is also a kind of biological clinical aspiration. From my perspective, the social pathways that I'm measuring are equally complex. and equally difficult. And you have to ask, why am I identifying? Because I think there are several pathways operating, so I try and identify them all, as we do in the metabolomics data and in the genomics work. But what this allows me to do is, in a sense, validate the pathways I'm interested in. If the things I think are early in the socioeconomic pathway lead to the biological change and lead to the distal phenotype that I'm measuring in the social science way, then that makes sense. It provides a very robust validation of what I'm doing.
Any more questions? Yes.
You mentioned towards the end when you were showing your data, there was evidence about vitamin E, and I'm just wondering, does that, do the data perhaps lend themselves to the idea that if you supplemented people, you could improve their frailty outcomes?
Yeah, I don't think this is really about interventions, unfortunately. So I don't think we're at a stage of thinking about interventions. So and as I've kind of revealed, I don't understand the biology very well, but as I understand it, what we found in this particular part of the metabolomics work is an important relationship between vitamin E and the carnitine pathway, which biologically makes theoretical sense. So thinking about the ways in which energy production might be really important. I guess is the way we then might think about it, and then think about how we might enhance energy production at a cellular level. Of course, it kind of relates to the previous question a little bit. It may well be that this is just the distal phenotype, and there's something earlier in that pathway that we need to get to. And that's where the other bits of the omics work that we're doing and that other people are doing is important.
James, I have a question about your concluding slides about your the way how you work, which is you work with experts across different fields, and how that contrasts a bit with how some social scientists or people in humanities might work, which is just the lone scholar type thing. Where, could you imagine ever as a lone scholar just working on biosocial research like this, or do you think it always has to be in conjunction with experts in different fields?
Sorry, before I answer your question, I just realised I didn't repeat the question that I was asked. So my previous answer was whether vitamin E provides an opportunity, the vitamin E pathway we identified provides an opportunity for intervention, perhaps through vitamin E supplements. You've hopefully got my answer. So the question that Trani has just asked me is about my working in a kind of interdisciplinary team with a range of experts and social sciences and humanities, often being lone scholar type work, and whether I could imagine doing the kinds of things that I do as a lone scholar. And I think there are kind of potentially three levels of answer to this. One is I've always done team research, so I don't think I've ever done research on my own, which might be something of a revelation, I don't know, but I've never done research on my own. I've always relied on colleagues, worked with colleagues over the years. And so that move into being a lone scholar, I think even when I write stuff on my own, I'm drawing on the stuff that I've done with other people. I don't write stuff on my own very often. So there is something about my personal ways of working that's important here, which does contrast with the ways of working of several of my colleagues in sociology and other bits of humanities. And I guess that relates in part to the kinds of work you're doing. So I think if you're doing kind of theoretically intensive work, trying to break new ground theoretically, then perhaps that does need more of a lone scholar thinking, perhaps with discussion, but more lone scholar type work and thinking. Whereas the type of work I do, of course, is complex empirical work, and that does require people who could do the kind of complex statistics, which I can't, who can understand the kind of different parts of the process that we're studying, which I can only get a partial understanding of, and so on. And maybe it relates in part to personality. I like working with people, and and that kind of intensive work with people is different from having just, which is also good to have, of course, the conversations over coffee every now and again. Yeah.
I was just interested in the use of ELSA and the sample data, and I thought social scientists are quite used to using those kind of large-scale sample surveys. And if you're working with medical researchers, do you find that there is any difficulties in that respect in terms of that they're used to work with different types of data, or do you need to kind of explain the kind of complex sampling that's taking place, and how does that kind of work in an interdisciplinary type of team?
Yeah, that's a kind of important. Sorry. So the question is how does our familiar use of complex survey data work with disciplines that aren't used to these forms of data quite often, I guess experimental data or whatever that might be, or small-scale sampling of clinical samples and things like that. And I think this is a really important question, particularly for the kinds of work that social scientists who are using large surveys, so epidemiologists, sociologists, social statisticians, political scientists as well to a certain extent, how the kinds of data we use are accepted. And I guess that there is that kind of importance of translating an understanding of the quality of the data that we're generating and the limits of the data that we're generating. So why do we want population representative data if those are the form of data we're using? Why do we want to use panel data if those are the forms of data we're using? Why do we use questionnaire data as well as perhaps measured data? How robust are the questionnaire measures that we use? I showed you depression very briefly. I didn't show you how we're measuring frailty. Are these robust measures? Do they have clinical relevance? Do they have biological relevance and so on? So there is that translational thing. And then there's the size problem. So the size problem for us is number of cases and multiple observations. The size problem for them is number of variables. And then when you put the two together, it's a kind of bigger problem. And so one of the things that the metabolomics team are proud of was that they were able to, and rightly so, was that they were able to do panel analyses of huge numbers of people. Now, for us, it's not just over 1,000 people isn't a huge number of people, but for them, this is a huge number of people. This is the biggest ever metabolomic study of its type. And yeah, absolutely, hours and hours, days, weeks, months of laboratory time to process these data. So for them, they then have to understand the value of having these kinds of data. So explaining how we need to compare frail with non-frail, why we need population representative data, why we are interested in change over time and so on. So I think this is a really very important thing, part of the collaboration is to illustrate the value of the data.
Thank you very much, James.
Thank you.
I now would like to invite Professor Gillian Bentley from the University of Durham to talk to us about a really interesting work that she's doing on epigenetics and social sciences. Thank you very much, Gillian, for coming today.
Thank you very much for inviting me, and apologies, I was late, but it was the trains which screwed things up a little bit. So yeah, I'm going to talk about how epigenetics can enhance social science research. And let me just get used to this. Yeah, so this is a slide that I often show my students, just out of humor, but basically to attack the notion that genetics are deterministic, and because I come from anthropology, whereas we're social scientists primarily. And instead, again, when I talk to my students, I like to give them this quote from Robert Sapolsky, who's an American primatologist, who talks about how genes don't cause behaviors, sometimes they influence them. And this is where I think epigenetics comes in really beautifully, because we can then merge our knowledge of genes and environment and talk about how we can use epigenetics to understand the interactions between the two, and also to talk about how they can enhance our knowledge of work that social scientists do. So for those of you who are not too familiar with epigenetics, what it does is it's a field of study that makes interactions, links between genes and environment, and those links form the resulting phenotype, that mix of genes and environment that people talk about in biology. It's a very old term, in fact. It was first used in 1942 by Conrad Waddington, And he was interested in developmental origins, so it's kind of rooted in our understanding of early life development. We need to understand too how epigenetics is really important in also influencing or playing its role in plasticity of various individuals and how they can change through the life course. And the other thing to remember from various studies that have been done, that we can't just talk about one single epigenome referring to a single individual, but epigenetics works on multiple levels within an individual. So we have to think about multiple epigenomes as well. Now, in terms of social sciences and the environment, there was a really important editorial that came out in 2012 in Nature, where it talked about how this epigenetics provided a really excellent opportunity for there to be interaction, debate between social scientists, and how we can therefore tackle, using epigenetics, the nature-nurture argument that has been raging for quite a long period of time. In fact, there's still an argument, I think, raging between geneticists and epigeneticists about the respective values of their subdisciplines, but that's an argument for another time. But the Nature article said this was a perfect time for reconciliation of the two cultures. So what I want to do is talk a little bit about studies that I'm involved in now that are trying to marry these perspectives of nature and nurture. The other thing that I should talk about is I'm an anthropologist, and anthropology is a fairly unique discipline in that for a very long period of time, anthropologists sort of come in different varieties, and it's not that uncommon to find biological anthropologists and social anthropologists or cultural anthropologists in the same department. It's more rare to have a really good dialogue between them, particularly in the USA, where it's been known for quite some time that often there can be arguments between the two about the respective value of their own areas. And this is, again, where epigenetics, I think, could make a really great bridge. But I will say that certainly in my own university, in my own department, we have fantastic dialogue, and genuinely there are biosocial studies that are going on and dialogue between people who come from different perspectives. But for those of us who are more biosocial, I think making that leap into a field like epigenetics, something more biological, is less of a stretch. So perhaps as an anthropologist, got something of an advantage because I was trained in sort of multiple approaches to the study of humans. So these are then my disclosures, having talked about anthropology. I am a biosocial anthropologist, so I can bridge the gap a little bit better maybe than others. I'm also a reproductive ecologist. That's my primary specialty. So I've been spending a long period of my academic life looking at reproductive function in different human groups. And I'm particularly interested in how the environment impacts on reproductive function. And within that, because of my anthropological background, I don't just want to focus on one or the other, the respective biological and or cultural sides of how the environment can influence reproductive function, but there are many different aspects of the environment, and we must include that social side to be able to fully understand various influences on reproductive function. But with those disclosures, anthropologists as well, I mean, I collect my own data for the most part, or nowadays I send out my students and postdocs to collect the data for me. But I have collected a lot of data myself in the past, and I have done lab work as well, so that also helps a great deal in trying to understand some of the more biological sides of reproductive work. So what I'm going to talk about specifically in terms of projects today relates to how the early life can impact on reproductive function. And I also look at aspects of migrant health. I've used as a model to look at environmental impacts on reproductive function Bangladeshis in the UK, so migrant Bangladeshis, and gone back to Bangladesh to get groups that actually still live in Bangladesh. And more recently, although I'm not going to talk about that here so much today, I've been running a larger project looking at the effects of acculturation, integration on Bangladeshi migrants who live in different parts of the UK, and hopefully there'll be opportunities to do more epigenetic work on some of our findings there too in the future. to sort of set the scene, though, about reproductive function and to set it in a context as well of social science and issues like inequality that James was just talking about as well, I wanted to show you this slide, which comes from studies of different populations around the world. And this is looking at average progesterone profiles across these different societies. Just get the little point. Ooh, hang on, not that one. This one, yeah. So this is a typical, sort of the shape is a typical progesterone profile where it's high in the second half of the cycle. This is a non-conception cycle, I should say. But the striking thing is just the differences in these average profiles. So these were collected over many different women, sometimes over the course of several months. But what it tells us is that depending on what kind of society you live in, your progesterone profile will look very, very different. And this isn't looking just, obviously within a society. And James does work primarily, in sort of one society, differences in socioeconomics. But this is looking at groups that have completely different lifestyles in terms of subsistence, nutritional availability, and so on and so forth. And the striking thing here is that the poorer the society, the more stresses they experience, like the Tamang here, who are agro-pastoralists in Nepal. Their average progesterone profile looks completely different from this one here, which is mostly from Boston, well-fed women, I think primarily collected actually from Harvard University, so, elite, highly educated, very well-nourished women, and then others in between. And then what's interesting here, sort of clinically, is if you look in a textbook, you'd see that this is considered to be the marker at which, if you fall below it, your chances of conception are very, very low. And when you see profiles like this, and you know that communities like the Tamang are reproducing, it really challenges our notions as well of what we even understand in clinical medicine about what's normal and what's not normal. So it, again, it raises questions about what are we looking at here? Are we looking at the impact of genes, or are we looking at the impact of the environment? So I've been interested in a lot of these questions for a very long time. And then more recently, I was doing some work, and I'll talk more about this in a minute, about reproductive aging in women and some of the hormonal markers of reproductive aging. And there's a lot of literature I found in different fertility journals, mostly coming from people who work in assisted reproductive technologies, so things like IVF, which talk about how there are racial, ethnic differences in women's ability to respond well, if you like, to IVF treatments, which is kind of disturbing because it could mean that then people working in these fields might make assumptions about whether women are good candidates for these kinds of technologies. So again, it raises issues about equality of access. What do we even understand by ethnic differences? And again, I'll get back to this, but I wanted to raise it as a question because it's something that I think we need to think about in terms of, are we talking about here about genes, or are we talking about the environment? So the question that I want to pose here, but I'm talking about my work and what we found is, can we use epigenetic mechanisms, can we examine them to explain how we can get really rapid reproductive changes in migrants, even within one generation? So I'm going to walk you through some data first and then talk about these issues a bit more. Okay, so background info to this. So since 2001, together with my students, postdocs, collaborators, we've run 5 projects looking at the issue of early life development, reproductive function, and then more recently trying to explain our findings. And as I said, we've been working with Bangladeshis as our model, and you may ask, why did we pick Bangladeshis? Well, there's several reasons that have been very useful in terms of doing later statistical analyses. So they're a multi-generational ethnic group with a very long history of migration to Britain, and that's important because we needed to get multiple generations to look at. They tend to come from one particular region in the northeast of Bangladesh, a region called Sylhet. So that's good because you can control for region of origin. You don't have people coming from lots of disparate different kinds of environments. They come from generally the middle class, so people with the means to afford to be able to migrate. Of course, once Bangladeshis get to Britain, because of exchange rates with their own currency, they tend to be on the lower socioeconomic status bar in the UK, but they do originate from upper classes in their own country. Once in Britain, at least the first generation tended to settle in distinct neighborhoods. So again, you can control for the region where they live. In terms of controlling for cultural aspects, the majority of Bangladeshi migrants in Britain are Muslims. And so you've got lots of nice confounders that you don't have to worry about in doing your analyses. I mean, you do have to worry about a lot, but some at least control for in some way or another. So in terms of contrasting environments between Bangladeshis and white British people who we use as comparative groups as well, what is it that's different then? If they're from the middle class, we know they're not poorly nourished, so there's nothing in that respect that could explain differences that we might find. People from Bangladesh who migrate don't tend to spend long hours laboring in fields, so they don't have high levels of energy expenditure. But what we do think might explain the differences that I'll talk to you about in a minute is that in Bangladesh, even if you are relatively high socioeconomic status, you are still exposed to a far greater number of immune challenges. We have health histories from people collected by questionnaires. They've been exposed to a greater number of infectious diseases than we tend to be in the UK. And there's not very good access to good medical care. So despite our complaints about the NHS, situation in Bangladesh isn't really any better, and sometimes you can't get medical care depending on where you are. And then poor sanitation. Sylhet in particular is subject to seasonal floods, so that raises the issue of exposure to diseases like, cholera and everything else that comes from floods of rivers that are not very clean to begin with. So in terms of the study design, in almost every single one of the studies that we've done then with Bangladeshis, we have multiple generations. For the first generation people, as I said, we went back to Bangladesh and looked at people still living in Sylhet. We look at adult migrants, and we classify these as people who migrated after puberty. So in most cases, that's the age of 16, the last age at which we recorded puberty in people. Then we look at child migrants who came before puberty was completed. Second generation Bangladeshis born in Britain to parents who had migrated from Bangladesh themselves. And then we always have a reference group of people of European origin, so in generally white British people who we pick from neighborhoods that are similar to where the Bangladeshis are living. So again, control over neighborhood. And what data do we collect? So as social scientists, we get all kinds of questionnaire data, so our subjects have to fill in, very long questionnaires, getting data about demography, basically, their immigration histories, health history, nutrition, lifestyle factors, all those things. We collect anthropometric data for weight, height, skin folds, biomarkers for hormone analyses, and depending on what study we did, that could have been saliva, blood, or urine samples. So it's a lot to ask of people, and it's quite labor intensive. So our sample sizes look small relative to, say, James's, studies, which are much, much larger, but we collect, quite intensive amounts of information from each individual. So then what are the results that we found from these studies? In our first study, which is looking at women who are aged 18 to 35, so this is women who are prime reproductive age. They should be sort of at the height of their fertility, and these are studies, by the way, on non-contracepting women, or at least non-hormonally contracepting women, so we have natural cycles to look at. You remember I showed you a progesterone profile before from different societies. This is what a progesterone profile looks like normally, and for financial reasons, we just looked at that sort of elevation here, which is the only thing that's really biologically meaningful. So this is looking at that here. But these are the different groups of Bangladeshi women. And I drew this dotted line because basically you can see the difference here between women who never left Sylhet, the red line, women who migrated as adults. And then here you have a mix of the child migrants, white European women, and second generation women. And there's, a significant statistical difference between these women. So these, again, averaging profiles across multiple women who took part in the study. And this led us to conclude that the childhood environment, depending where you grew up, where you spent your childhood, seemed to have an influence on levels of your reproductive steroid hormones. And then with the child migrants, we split them up according to when they came to Britain. And if you migrated before the age of 8, your levels were much higher than if you migrated later on, although, you know, on average altogether there was still a difference between the adult migrants and the child migrants. So we were thinking that maybe 8 is something of a threshold, and more on that in a minute. The other thing is, from our data, we were able to calculate rates of ovulation between groups. So these are the rates. We also had estrogen data, which I'm not showing you here. But from combined estrogen progesterone profiles, we calculated rates of ovulation. And if you were born in Bangladesh, basically this was your rate of ovulation per cycle, and this is for European women. So you can see again, it's very different. If you're born and raised in Bangladesh, you will ovulate less frequently in a cycle than a white British woman would. And then this is recalled age at menarche, first menstruation in girls. And the child migrants had the youngest age of menarche of any of the groups. And the women who had grown up in Sylhet had among the latest. So what was interesting is our white women, we're not that dissimilar from them. But what's important to remember here is that the migrants who'd grown up in Britain seem to have a sort of accelerated age at puberty, which is interesting. And then we did a second study. This is looking at older women, aged 35 to 59, and we were tracking them to look at the rate of reproductive aging, and we looked at a suite of hormones that tell you about reproductive aging, the number of eggs left in the ovary, and also we asked women who had gone through the menopause already for a recalled age at menopause. And we did a probit analysis on age at menopause, and you'll see here that the age at menopause for women still living in Bangladesh was about 48 years, migrant Bangladeshis 49, and the white British women close to 53. When we looked at a suite of hormones that tell you about ovarian aging, and I'll just mention them here. This is anti-Mullerian hormone, inhibin B, and follicle stimulating hormone. As you get older, AMH and inhibin B go down, and FSH, follicle stimulating hormone, goes up as you approach and go through the menopause. But again, what's interesting is you can see here these dotted lines, depending on where you grew up, the levels of these hormones and the age at which you were reproductively getting older was earlier among those women who had grown up in Bangladesh. So again, a second study that seemed to confirm what we were finding earlier about the influence of the childhood environment on reproductive function. Then also we've done another study. This is one of my students now, a postdoc somewhere else, Keson Magid, who worked with Bangladeshi males. And he did a similar study to what we were doing in women, but with a wider age range of men, looking at how the childhood environment might influence levels of salivary testosterone and age at puberty among these men. And he found things that were, remarkably similar to what we found in women. Here's that dotted line again. Where did you grow up? If you are a high-status white man, or you're a second-generation Bangladeshi or a child migrant, your levels of testosterone are higher, significantly higher, than if you grew up in Bangladesh, or interestingly enough, are a low socioeconomic white British male. And again, age at puberty, this is recalled age at puberty using a suite of markers for males, because you can't ask them about age at menarche, of course. But using that suite of markers, again, similar findings. These are color matched as well. That if you were born in the UK, so these are second generation, your age at puberty is significantly earlier than if you're born in Bangladesh. So nice confirmatory findings among the males. And again, this is looking at levels of testosterone, salivary testosterone in men, depending on when they moved to Britain. So as a child migrant. So this is morning and evening testosterone. That's why you've got these two lines drawn, so AM, PM. And you can see, again, differences between the two. So again, this sort of threshold of 8 might be interesting. To examine that, another one of my students who's now at Columbia University in New York, Lauren Houghton, she looked at adrenarche, which is this really interesting threshold period of time in middle childhood that usually occurs between the ages of 5 to 7 years in kids. And it's a time, James talked about this hormone DHEAS, We were measuring that in the children. It's an important marker of adrenarchy, which used to be thought of as a precursor of puberty, but now we tend to think of it as being a marker actually of cognitive function, because DHEAS is really important for completing processes of brain maturation. But what Lauren found is that if you were a first-generation girl who moved before adrenarchy to Britain, you experienced adrenarchy as measured by threshold levels of DHEAS 2 years earlier on average than any other group. So there's something going on about this period that seems to be important. So we have very consistent results across all of these studies. I think we can conclude from these consistent results that the childhood environment seems to have some influence on reproductive function. I'm sure genes operate, obviously, on some things like age at menarche, but this would seem to indicate that when you're talking about ethnic groups, comparing Bangladeshis to British people, you can't say from these results that, genetic influences would be more important. Pre-adrenarchy seems to be a kind of threshold, but we still see influences across the entire prepubertal period. So, seeing these results for a very long period of time, I was really interested in how can we explain this? What is going on here? How do we get at these very rapid changes in what is happening in these different groups? Some years ago, one of my students did a little pilot study with a colleague, Martin Wichwend, at UCL, where we did a very small study with 20 women, comparing 20 women in each of these generation groups, comparing first and second generation women. And we looked at methylation, looked for differences in methylation patterns in particular steroid hormone receptor genes. So I showed you progesterone profiles before. And we looked specifically at the progesterone receptor gene. And we did see in this very small study significant differences in the methylation patterns between first and second generation women in this particular gene. Small study, though, hard to get funding, as James mentioned, to continue this kind of work. So that was sort of where we left it. And we felt too small of a study to actually even publish, just a little pilot study. So then, a few years ago, ESRC, BBSRC had a call to try and get social scientists and epigeneticists to work together to look at questions of interest to both parties. So I went to their sort of scoping meeting and found there a couple of colleagues, Reinhard Sturger, who's in the University of Nottingham in the School of Biosciences, and Philippa Melamed, who's at the Technion in Israel. And that's really nice because it just does show, that we can also get international collaborators as well for these studies. And what we are trying to look at, this is the title of our study, Epigenetic Stability in a Stressful Environment and Its Effects on Reproductive Function. And what we're trying to do here is look for evidence of epigenetic changes, in particular genes that are, if you like, sort of at one of the higher levels, controlling the release of other hormones that I've looked at a more basal level. So we're looking at this hormone called gonadotropin-releasing hormone, which is released from the hypothalamus, but it controls the cascade of other hormones, even down to the ovary, where we get our reproductive steroids like progesterone. And in this study, Philippa Melamed, my colleague at the Technion, she works with animal models. She works with mice specifically, and we've set up a model. She's set up a model looking at early life stress in mice. So she's trying to mimic the potential effect of the immune stresses that we think Bangladeshi women are experiencing in Bangladesh. And also, of course, she's got control groups that are living without that kind of stress. So we're trying to replicate what we see in the Bangladeshis in mice, where we can obviously harvest tissues from the mice that you can't harvest from humans for ethical reasons. And this is kind of a bit distressing because I used to be able to say that no cute furry animals were ever harmed in the course of the studies that I did as an anthropologist, but I can't say that anymore, although of course all of the studies conform to the higher standards of animal care in the lab. The other thing that we've done is collect buccal swabs from Bangladeshi women, from first and second generation Bangladeshi women, for several reasons. First of all, we wanted to see if we could see any differences in methylation patterns in particular genes of interest. And the other reason why we did this is because there's a lot of discussion in the epigenetic literature about what tissues lend themselves to analyses, so specific epigenetic studies. And although it's possible, for example, to collect things like vaginal swabs from Bangladeshi women, you wouldn't necessarily want to do it unless you had to. So one of the things we're going to do is we also have buccal swabs from the mice. We can get vaginal swabs and ovarian tissue as well. So we are going to do a little study sort of comparing results using the mice as well about which tissues lend themselves to analyses of this kind. And then finally, my colleague Reinhard Stöger is looking at epigenetic stability, whether you can look at stress across the life course and whether in our samples there's indication of this early life stress in the way in which epigenetic changes occur, the pace at which they occur across the life course. So very quickly, because I know I'm running out of time, this is what we found so far, and the study's still ongoing in the mice. In the treatment mice, age at puberty is later, so that looks very similar to the Bangladeshi women who grew up in Bangladesh. We can track here as well weight changes, which are more visible in the treated mice. And then here, this is looking at a number of genes and their gene expression relative to control. So the gene expression in the treated mice is lower. As you can see here, this is the progesterone gene, luteinizing hormone, and follicle stimulating hormone. And these hormones here, they are responsible for the production, if you like, of the ovarian steroids, including progesterone. So some nice results here that seem to sort of mimic what we see in the humans. That's where we are so far with that. And then Reinhard has done some studies using methylation epic bead chip arrays, where it's sort of an epigenome-wide studies using data from the Bangladeshi women, and we are already picking out differences between them. This second graph here apparently proves that this is not a batch effect, it's a real effect. Like James, I don't know all of the technicalities involved with this. And one of the things that we found, this is a very complicated slide, but the basic message to take away from it is that among the sort of clearly significantly differentially methylated genes between the first and second generation Bangladeshi women that we found differences in this hippo signaling pathway that in studies of other species have been shown to be really important in terms of ovarian function. So we're at the early stages of analysis, but that's intriguing and it's very encouraging as we continue with this work. And again, we don't have results yet from whether buccal swabs are really good tissue to look at, although these data initially are encouraging because the last slide that I showed you came from the data on buccal swabs. And we're still waiting for results about analysis of epigenetic stability, so ongoing, as I said. So watch this space. And then finally, I just wanted to throw out a few further thoughts about social sciences and epigenetics, and what can we get of value of this other than some nice explanations for those of us who are interested in biological mechanisms. So some of the further thoughts about this is, from the biological perspective, the fact that humans and other species are highly plastic, I think, is a really interesting finding in and of itself, because when you're looking at the effects of the early environment, it immediately should remind us that cultural aspects, adverse environments, can have a really marked and early effect on individuals. The other thing, specifically about reproductive function, is that given this plasticity and the effects of environments, there's a number of trade-offs we need to think about, resulting in inequalities, such as in that first slide I showed you about progesterone profiles, and risks, health risks, that we need to think about. And as I said, I showed you this slide before. Our findings raise issues about, why is it, given differences between environments, poverty in some parts of the world versus others, you get women who can look so different, and what does that mean for potential reproduction? And then specifically to do with populations, South Asian populations, including Bangladeshis, in terms of trade-off risks, what happens when you migrate and all the rest of it. In terms of health implications, we know, for example, and this is just looking at odds ratios of whether women of different ages, South Asians, will, in their own home countries, whether they're at risk for breast cancer or not. The odds ratios are lower if you're a South Asian in your own home country. Once you move to Britain, this is just a slide from one study that shows how your risks actually go up. And in terms of breast cancer, your risks for breast cancer depend a lot on lifetime exposure to higher hormone levels. Our studies have shown that if you are a Bangladeshi woman and you move to Britain, your hormone levels will go up. But then, you know, there's a bunch of cultural factors that affect breast cancer risk too, including breastfeeding patterns, and we have data from our studies about what happens to breastfeeding patterns when you move to Britain. A woman who lives in Sylhet will breastfeed for about 18 months, but then if you move to Britain, you stop breastfeeding essentially at around 6 to 8 months. So again, these are changes in cultural patterns, and we have to think about these cultural, social factors as well as the biological when we're thinking about health risks. And then for males, prostate cancer rates generally are thought of as being low in South Asian men, but Kesson, who did the study on testosterone, collected data from the International Prostate Symptom Score questionnaire, and here you have child migrants and second-generation men, and you can see that their symptom score goes up dramatically once they get to Britain. So again, health risks will change. You know, risks change for migrants, and we need to think about these things when considering the health of migrant groups. And then I mentioned to you before, I showed you that slide about these studies that have come out from the assisted reproductive technology people about how South Asians have lower success rates for things like IVF. Their claim is that this is an ethnic or racial difference. Well, we've pointed out in our publications that maybe it's got nothing to do with sort of genes, racial aspects, but it might have everything to do with development and environment. And again, I think these things are really important in the way people are treated in clinical settings. So just to conclude, I think from, you know, at least what we've done and the promise of epigenetics, combining social science and biological data, is that we can do a lot. I mentioned here, you know, anthropology, sociology, but there are a lot of people working in human geography too, who I think do work that's quite similar. The other thing I want to point out is that our studies are very targeted, and there are lots of people out there doing sort of more general epigenome-wide studies, but I think there's a place for targeted studies asking very specific questions. I'd like to see more of that. There's also value in doing cross-sectional studies, as we've done for the last 12 years. You know, they're cheap, they're quick. I'm not trying to, you know, diss longitudinal studies, of course not, but, you know, there's a place for cross-sectional ones too. We need to remember, though, the field is still emerging and developing, but we definitely need more collaborations across social sciences and biology, and more money for these endeavors. So, thank you.
Thank you very much, Gillian. We have some time for questions,
Okay.
if that's okay. I just want to invite those people that are online also. Please send in your tweets, any questions. We've got a comment saying, thanks for the streaming, strong case for interdisciplinary working. So those of you online, just tweet away at NCRM UK. But questions for Gillian? James?
Can I ask a slightly tangential question? We touched on it, which is the kind of tissue-specific epigenetic work. And when I hear about epigenetics, I don't do epigenetics. When I hear about it, people are always arguing we have to do some tissue-specific stuff. But I guess I'm guessing from you that may not actually be necessary. We might see the same processes across different organs.
Yeah, so the question is, I'll repeat it. Yes, the question is, you know, do we really need tissue-specific studies in order to ask questions that may be related to specific genes, cells, parts of the body, etc.? This is a really interesting question, and yeah, people do talk about that, and that's one of the reasons why we wanted to do comparisons, partly using the mouse model of what the buccal swabs will tell us, the vaginal swabs and the ovarian tissue. When we do comparisons, looking at specific epigenetic methylation patterns, are we going to see the same patterns? We're going to get the same sort of richness of results. We don't know the answer to that yet from this study, but that's why we were interested in this, because obviously if you're working with humans, you can only do so much invasive work. So that is a question that I think really does need to be answered, and hopefully our study will go some way towards that, at least on a limited scale.
Gillian, I had a question about you working with animal models and also working with human subjects. And you talked about expressing mice.
Yes.
How the hormone levels and so on. Very different. But to what extent can is stress in mice comparable to early life stress hormones, validation, women? How do we know? It's
Yes.
to know what
Yes.
is stressful, but how do you
Reconcile
get
that.
from an
Yeah.
animal model
Yeah.
stress?
Yeah, so the question is, how can you make inferences from animal models that stressing out the mice to real life situations in humans? Of course, this is always a problem when you use any animal model, I suppose. And the stress that we were trying to induce in the mice was using a chemical, which was trying to induce the potential stress that people would be undergoing if they were immune challenged. So this was specifically trying to replicate what we thought was going on with the Bangladeshis. It's never going to be exactly the same because we're not allowed to infect, for good reason, these mice with specific diseases. So you can only use a sort of analog. We try and get as close as possible to what we can do given the constraints of using animal models. So yeah, it's not exactly the same, but it's the best that we can do.
It's not like you give the mice a questionnaire to fill out.
No, that's right, yeah, and you can't ask mice to fill out questionnaires as well. Yeah, limitations of the animal model there too.
I'm really fascinated by the graph you showed with the progesterone levels across the different societies. So you gave a little bit of an explanation about that, but is it largely nutritional or?
Yeah, so the question is about what explains those massive differences in progesterone levels between different societies. It depends on the society, but I did work with one of them, the Lesse, who were a slash-and-burn horticulturalist group in Central Africa, and there it was everything. They were nutritionally stressed, the women worked incredibly hard, physical labor in fields and so forth, and there were a number of tropical diseases that they were exposed to across the lifespan. So they had, everything that you could possibly imagine. There were also some Polish women who were an agricultural society but not nutritionally stressed, who were slightly lower than the Boston women. So I think you could actually probably make a gradation according to the stress and pile multiple stresses on, and you'll get the worst looking profiles. But what's interesting is that these profiles seem to be typical to that population. And as I said, it raises questions about our understanding of what's normal. Maybe a lot of people think that Western populations, that we are the abnormal ones with our very high hormone levels and other functions.
Well, any questions? If not, thank you, Gillian, once again.
No, thank you. Thank you for inviting me.
So we're going to we'll be taking a short break now. We'll come back at 3:30 or so.
Thank you.
think that's what the red pointer.
Yeah, I'll find a way to disable it somehow.
Okay, thank you everybody again for coming back. I'd like to introduce our last speaker of today, Professor Melinda Mills. She's the second of our speakers who's traveled from afar to come and speak to us today. So thank you very much, Melinda, for coming today. And she's going to talk to us about social sciences and molecular genetics.
Okay, thank you very much. I think that my talk will actually bring together the last two talks. There'll be a lot of overlap and similarities. So I'm a demographer and a sociologist who entered into the foray of molecular genetics within the last couple of years. At first, I think it's important, as everyone does, but to acknowledge two things here. One is that I don't do this alone. So I work, as Tarani said, not as a lone wolf, but also as James, I work with a team. So everyone has helped in this, but you should also have, this is my Brexit slide as well. So you should also be aware we were able to recently recruit one English person, Charlie. So, but that's always something that we have to keep in mind during this process. So what do I mean by genetics? I think that, some of you have already covered this, and I wasn't sure what the level of the audience was, so you can feel empowered for the next two slides if you know what I mean by this. We have looked at behavioral and molecular genetics, and what I'm going to talk about today is in the realm of molecular genetics. So I think some of you may be familiar with twin studies in behavior genetics, where you compared, the percentage of traits that were attributed to genetic, shared family, or unshared environment, which could be your spouse or your workplace, and aspects such as that. So these are these heritability estimates that everyone often hears discussed. But what I look at is molecular genetics. And this is where, we differ on these locations on the genome, and that's referred to as single nucleotide polymorphisms, or we often refer to them as SNPs. And then what we do is we try to isolate them, and then we look at their biological function and structure. So very similar to what the other speakers were doing. And you've probably seen this, we're looking at, this is SNP heritability, so, but it's different, as you'll see in a moment, from some of the twin levels. But you'll see that, some of the more anthropometric biological traits, such as height, eye color, freckle count, these have been shown to be quite a bit related to genetic variation. But when we get into the more complex behavior, what you can see is that we have quite a difference in terms of what we can predict and what the genetic prediction would be, but it's also lower levels. So this comes into more of an area where there's been different studies that have shown different results across different populations. So the topic, or the question was, can social sciences ignore genetics? And I'm going to show you the yes side and the no side. And I think there are some things that we can really ignore. I think we can ignore a lot of the previous candidate gene studies. There have been some new ones, but I think that the geneticists, when they talk about social scientists and why they're a little bit worried about social scientists entering into this domain, is because of these candidate gene studies. So these were early studies conducted 10 or 15 years ago, and they were easily conducted often on a few genotype markers, which was largely cost-related, and they were based on some of the animal or mouse models that we were talking about before. And the problem with these studies is that they generated a lot of bad research. So you might be familiar with some of the studies by Beaver and others that found this gangster gene or the human warrior gene. And all of these studies, that are present in around 40% of the population, they were publishing these studies quite early. And this is what, when I was first starting to work with molecular geneticists, they were very worried about. That people would be looking at distal traits that weren't that related to biology, and that they would be looking at these sort of sensational findings that were definitely the case with these early candidate gene studies. So I think many of you might be familiar with these, but there's been sort of countless articles now coming out saying that most of these candidate gene studies could not be replicated. So this is from Duncan and Keller, but there's other studies, you know, just showing that it was very, often these were done on very small populations, on selective SNPs. They rarely replicated. They wouldn't have the statistical power to actually show these effects. And there was a very strong publication bias in publishing, you know, that this gene has an association or has an effect and is related to a certain behavior. That led the editor of Behavior Genetics in 2012 to say something what I find incredible, because I've been an editor of a journal too, that says it now seems likely that many of the published findings of the last decade are wrong or misleading and have not contributed to real advances in knowledge. I mean, it's incredible. So this field has really moved quite quickly, and as social scientists, it's often hard for us to keep up. And for that reason, it's really important that we engage very actively with molecular geneticists or biologists, because the field moves very quickly. So these articles were published in the American Journal of Sociology, in population studies, in, you know, very high-level journals, and most of them have not been able to be replicated. So now there's a demand for adequately powered studies that can be replicated across different populations. So that's some of the stuff that I think most of us would agree we can ignore. But why have we ignored it? And I think there's been a lot of discussion about this. But sociologists and social scientists in general have quite an allergy in some ways to genetics. And that's related to things such as the bell curve that I'm sure many of you have heard, that they talked about inequality and stratification as based on genetic endowment, and that meant that any sort of social policy, you know, you couldn't intervene, it was as it was. And I think that's been a real problem that's plagued us. But, you know, I was amazed last year, I gave the keynote at the German Sociological Association. So in Germany, so imagine on genetics and sociology. So things have moved on, I think, and people are much more embracing of the topic. But I think a real issue, and this is why we have this NCRM project that we're working on, is a lack of interdisciplinary training. So that's some of the stuff I think, you know, that we can ignore, and that's why we ignored it. But of course, I'm here also to talk about some of the opportunities as well. And I think the second part of my talk will be about why it could be dangerous to ignore it as well too, and what I've learned by entering into this area. So I did this research for most of my career. So it was very socially deterministic. I thought that a lot of the behavior that I studied, labor market, fertility when you have your children and how many children you have, and employment, I thought it was related to, you know, choice, individual characteristics, and the social environment, you know, educational systems, housing markets. And I think that's what a lot of social scientists look at. But then I went out, for a few drinks with some of my genetics and biology friends, and they would laugh at me, and say, oh yeah, this is Melinda. She studies fertility, but she doesn't think it's biological, and it did actually sound a bit absurd. When you thought about it, you thought, okay, I really do have a blind spot. So I think that, there's and as we saw from Gillian's talk, we have the social environment, we have choice, but biology also plays a very strong role as well. And we've had a blind spot in many ways in the social sciences. So we summarize sort of, if you're interested in some of the designs in a PNAS article last year, where we talk about some of these different designs, and James talked about them as well. So if you're interested, you can look how we went from this classic design, looking at twins and families and GCTA and Gremel models, to genetic profile scores. And that's kind of the journey that I've taken, and I will also just walk you through it now on some different topics. So some of the relevant techniques and ways that we've been approaching molecular genetics as social scientists have been looking at things such as Gremel, and I'll show you that in a moment using GCTA software. We've conducted several genome-wide association studies, and as well, what we produce from those those GWAS studies are what's called polygenic scores. So it's aggregate or a single risk of genetic risk related to a trait. And I'll show you about how some of those work. So in 2015, we had done some twin models. I'm not going to show you that, but we realized that it's quite clear that there's quite some genetic relationship with fertility. So what we look at is age at first birth and number of children ever born in many of our studies. And what we did is after we conducted some of the twin models, we realized, okay, but we have whole genome data, so we can actually look at individuals who are unrelated and look at the genetic relatedness between them. So that's this GCTA or GREMO models. So James and I might be 10% related, and Gillian and I might be 50% related. And you can look at people that are unrelated in a data set and look at their genetic relatedness and see the relationship to the phenotype or outcome. So as I said, we looked at, we call it fertility, but to make it cross-disciplinary, it needed to be human reproductive behavior. And we looked at age at first birth and number of children ever born for individuals that had reached their reproductive, end of their reproductive period. And what we found was interesting. We found that when we looked using these methods, that the heritability or the SNP heritability or the genetic heritability of age at first birth and number of children ever born was 15%. So 15% of when you have your first child is explained by genetic variation, and it was 10% of number of children ever born. This is really interesting considering we as demographers and sociologists had only looked at social factors until then. So it looked like we were missing around 10 or 15% of the explanation. But then, of course, a lot of people asked, and we thought about it as well, okay, so it's 10% or 15% genetically related, but you're back to the same point as the twin study. So if something's heritable or genetically related, you kind of wonder, what are the genes and do they have a biological function? And this is what James and Gillian just discussed as well, too. So in order to do that, we conducted what's called a genome-wide association search. And this just shows the different chromosomes and it shows for different related diseases the different markers that have been found. There's about 3,000 studies. So on the different chromosome 3, and we have one of these dots, or 13 actually, now in terms of these studies. So I'm going to give you some examples of GWAS studies that social scientists have conducted. So I'm involved in this one as well, and that came out last year in Nature Genetics, where we found genetic variants associated with subjective well-being, depressive symptoms, and neuroticism. You might have also been aware of the educational attainment GWAS. So there was one published in 2013 in Science, and then in June of last year in Nature Genetics, they also found 74 loci related to the number of years of education and obtaining college. I'll show you some of the results. They're redoing the study, and it's really exciting. They found about 1,300 genes. I just saw them present it last month, and that's under review at Nature or Cell or something, I can't remember, but that one will be coming out very soon as well. And what we did is we also published last October in Nature Genetics, where we found 13 different genetic loci related to reproductive behavior. And what these GWASs do is it's basically data mining, what sociologists call data mining and what geneticists call hypothesis-free research. And you go across these different gene variants and you regress across all of the different SNPs on your outcome or your phenotype. So in our case, age at first birth, a number of children ever born. And because of all these regressions, you have Bonferroni correction and very low p-values. You have to mandatorily replicate it now for the reasons I showed you before. And you have 2 quality control centers. So we do the analysis in one center, and an entirely independent group does the analysis in the other center. So that gets rid of these kind of false positive findings. It'd be great to do that in social sciences as well, too. You have to pre-register your analysis as well, so you're not phishing. So you have to pre-register everything you do on Open Science Framework. So our sample size is one of the top 10 of the 3,000 largest GWASs conducted. These are the total sample sizes for age at first birth and number of children ever born. And it was a combination of 63 data sets. And we combined it from scientific studies, but also commercial companies and insurance companies. And although I didn't collect my own data, I can tell you getting these data sets was probably just as time intensive, getting them particularly from the commercial companies. And I think what's really exciting is we had a very large sample of men. So what we found, this is what's called a Manhattan plot. These are the different chromosomes, and these are the areas of significance. And this is suggestive to the -6 and to the -8 means that you have you found a genetic loci or a hit. So what you can see here is for age at first birth, we found 10 significant loci, 1 in women only, and for number of children ever born, we found 3, and 1 was in men only. And I'm not going to go through in detail, but what we do then as the demographers or social statisticians is we produce, these analyses, which for us is possible, but then we hand these over to the biologists and the molecular biologists to work on some of these different analyses. And they did a series of different analyses in order to see, okay, what is the causal function or role? Is there enrichment? What's the causal pathway or gene expression? And I won't go into that in detail now. But it was interesting, some of the things that they did find was, some of the genetic loci that we located were related to sperm function, quality and function in men, infertility, hormones. That's why I was really excited about the last presentation as well, too, and about estrogen, FSH levels. And there were some very clear overlaps for women with endometriosis and polycystic ovarian syndrome, which are related to infertility. So it was interesting, these behavioral phenotypes were having a very clear biological relationship as well. And the lead genetic loci in chromosome 3 was linked to methylation and expression of genes known to play a role in sperm function. So that's something that we're now working on with CRISPR and some zebrafish follow-ups as well, too. So what's coming soon? So this is also a bit depressing if you're a social scientist, but what we did last October is actually old news. The methods have advanced and the sample sizes have advanced. So we're now redoing everything. The postdocs just went, yeah, we're redoing everything again. And we're now looking at 4 phenotypes. Number of children ever born and age at first birth, but I'll show you in a moment, we're looking at childlessness and age at first sex as well because of the overlaps. We're using different kinds of more densely imputed data, and we're looking at the X chromosome. So our new sample is almost 1 million for childlessness and number of children ever born. And for age at first sex, it's actually fairly large, and for age at first birth. I won't show you the results because we're just finishing the meta-analysis now, but now we're up to, in some of the phenotypes, around 300 different genetic loci and a very well predicting polygenic score. So let's move to those. So then you do your GWAS, and then you produce what's called a polygenic score, because it's never for our complex behavioral phenotypes such as well-being or education or fertility. It's never one genetic loci or 2. It's usually a combination. So what you do is you do the combination of the and you produce a polygenic score. So you have to think about it like a question about attitudes about immigration. You ask 10 questions and combine them. It's a little different, but it's similar in terms of you can have one measure to measure the genetic predisposition or risk to a certain outcome. So in terms of the educational attainment polygenic score that was published in Nature, the old one that was published in Nature last June, they were predicting, including all the SNPs, which is standard, they were predicting around just over around 6% of the variance. And that's with a GWAS study, so around 6% of why you obtained the level of education that you did was related to some genetic basis. And this is their new study that's under review right now, and it's really interesting. So this isn't published yet, but if we look at the newest GWAS in education, and you compare it with our standard social science predictors for levels of education, you see that, parental education was, explains a lot. So this intergenerational transmission of educational behavior explains a lot, as does the mother and father's education and cognition. But they're actually, with their genetic score, explaining quite a bit. So we also wanted to look at, in our study, well, what's the predictive power? So, we spent 4 years on this. I got 63 data sets. It's about to get depressing, by the way. And then we wanted to see, what is the predictive power of this genetic score and these genetic loci that we found. What you have to do is you have to replicate it and show that this has some predictive power out of samples. So we looked at various studies in the US, Netherlands, Sweden, and the UK. And what we found, remember I showed you the Greenmuller GCTA SNP heritability models. They were saying that we would find about 15%. Well, we found, including, and these are the different data sets, that age at first birth, our polygenic score predict about 1% of the variance. But if you only add one social science variable in, one of the usual suspects, that actually is a low predictor as well. So things are often multivariate. But it predicted around a half-year delay in women and 4 months in men of when you're going to have your first child. This is my son, and this is number of children ever born. So you can see it predicts, there's no other way to graph this, but it predicts about 0.4% of a child. So if you try to think in terms of effect size. So when it becomes more interesting is when you try to understand, okay, so these polygenic scores, these genetic loci that we found, are they related to other traits? And I won't go through these, I promise, all of them. I'll just pick out a few. But what we did is we looked at all of the related GWAS studies that we thought, and we wanted to do something called LD score regression. And you can now all do this online, but last year you couldn't, but now you can just type in everything online and see its genetic relatedness. We wanted to see, so age at first birth is red and number of children ever born is blue, and the star means it's significant. So we wanted to see, what's the genetic overlap between some of these traits and our traits. So this looks at age at menopause. We can see that there's a relationship, and I'll talk about that in a moment, but we can go down to some psychological or psychiatric, height, BMI, different factors. So what we found, and I'll just zero in on this, is there was a link with human development, and that's why I was really, the talks, there were so many similarities with them. And when we looked at LD score regression, we really found that there was a link. So this is age at first sexual intercourse with age at first birth. So later first sexual intercourse, or genetic propensity to later first sexual intercourse, later age at first birth, genetic propensity. So there's an overlap. But as well with voice breaking in boys and age at menarche and menopause. So this was something that we thought was very interesting. And it seemed that a later age at first birth was linked to later timing of puberty and development. And indeed, we're following up this study now, and we're finding that there's a genetic overlap with certain individuals where we have a shifting of menarche, age at first sex, age at first birth, to some extent menopause, but menopause doesn't seem to budge very much. And we thought, could this be linked to longevity as well? And just when we were about to publish it, of course, someone else did. But we're still following this up. And actually, this just came out in PLOS Biology last week, I think. And they're using our polygenic score to predict mother's age at death. So there seems to be some relationship between later genetic propensity to have later births and shifting of the reproductive window with longevity. So I talked about the polygenic score for age at first birth, and I gave you some examples of predictions related to longevity. We wanted to see if it's the polygenic score for the number of children you have, if that's related to other factors, and I'll give you the example of childlessness. So what we did find is one increase in the standard deviation of the polygenic score for number of children ever born was related to a decrease of around 9% in the probability to remain childless for women. we've replicated this now in some American studies too, and we're finding some significant results for men as well. And then the other obvious thing that you start to think about is differences. As a sociologist, you're always thinking about these, and James talked about this, these differences between groups. So in July of this year, we published in the European Journal of Human Genetics, we tried to look at sexual dimorphism, so differences in men and women, because the question you get asked the most on this topic is if infertility, or infertility is if it can be transmitted, how is it possible to transmit genetically childlessness? I mean, this is the question you get asked. And what we were looking at is it appears that there's sexual dimorphism and that the genes contributing to male childlessness can be passed on via male lineage and vice versa for women. And we're now looking at intergenerational data and indeed seeing which genetic loci are being passed down. So if you're interested in that, you can look at it further. But the punchline there is that we started to look at these individual genetic differences and saw that there were differences by men and women, which isn't done very often in these genetic studies. Unless it's related to a really female or male trait, often the populations are just grouped together and it's not really looked at whether they're male or female. And I mean, I had to fight for this too. The geneticists were like, well, why would you look at differences between men and women in relation to fertility? And, I was thinking, well, if you've ever had childbirth, you know that there's a difference, there's really a difference in the fertility process. So we found preliminarily that there's different sets of genes related to fertility for men and women. So when we have these factors, I was saying they predict maybe 1% or 2% of the genetic variation. But what about when you include other social factors and look at it in a multivariate model? We've looked at this in the health and retirement study and looked at it in the Wisconsin longitudinal study. So these are American studies where you can easily get the access to the data, around 10,000 cases each. And we wanted to look at, the relationship between genetic and social factors in predicting childlessness, because we've had an increase in childlessness. It's U-shaped. It used to be very high around the 1900s, and then it was very low. It was around 20%, then it was around 10% in many societies, and now it's up to around 20% again. So we wanted to look at this across time, across different countries. So we know that infertility is related to a lot of genetic factors. So for females, it's related to PCOS and endometriosis, and for men, it's often related to sperm quality and function. So we gathered the genetic findings that we could find that would predict childlessness or infertility. We looked at those. Some of them are not available, and these people were kindly gave us their results. And then we looked at the social factors that you often look at when you're looking at childlessness, so age at marriage, educational attainment, occupation, these kind of factors. Sorry about the size, but the main thing is when you include them, this is for women and this is for men. This is for the Health and Retirement Survey, but it's replicated in the Wisconsin longitudinal survey. And you find what you would expect, that education is a predictor, of childlessness, as is, some of these occupational factors. We know this. But we find that if you have the polygenic score for having a propensity to have children later, that seems to play a role as well. When you include social factors as well, so the genetic factors aren't entirely explained away. And for men, definitely the even though it was a small GWAS, sperm defects were very important, as were these markers that we had found. So what can the social sciences add to genetics? So what, the explanation, I think James talked about it a lot too. Once we started to get into it, we started to realize that we were looking at very basic things, such as talking about, well, there must be a difference between men and women. And then what Gillian discussed, these differences in progesterone levels across different societies, we were also thinking the same thing, because we combined 63 different data sets with all multiple cohorts across different countries and periods, and we assumed that they were all the same, which for a social scientist is a big leap. So I think what we can, add to this is looking at the social structure, context, and time. But James talked a lot about the expert measurement that we have too. So that became very clear once we started working in this, that we really had something to contribute. And also attention to group differences became very clear, that we were looking at things in a very different way. So this just shows all of we did sort of a summary of all the twin studies that looked at heritability at age at first birth, the number of children ever born. And I don't want you to go through all of it, but what you should see is, wow, there's a big difference across countries, and there's a big difference across birth cohorts. So that's what we found. We kept seeing this. There's this huge variation across country in both birth cohort, but all these genetic-wide studies that were being published were not taking that into account. And if you look at, this is age at first birth from 1920 to 1980, so people that were born during those periods, you'll see that there's a huge variation across time, but also across different countries. So it's similar to what Gillian was showing about the progesterone levels. But this is Australia, Estonia, the Netherlands, Sweden, UK, US. You'll see that these ones were having their children at age 30 as they are now, but here it was low. So there must be differences across different birth cohorts and also across different countries. And if I think about the social environment, this is my mom, and this is me. And this is my mother-in-law and her grandmother. You can see that wearing eyeglasses is definitely heritable in the family. But look at the different in social environment. I mean, it's just a stupid example, but of when we're having children. I mean, look at the social control is just in the room, about when she should have a child, how many children she should have. And look at me, I don't know, I'm somewhere in the Netherlands watching a marathon or something. There's really, I had a lot more choice in what I did and when I didn't. As a result, I think that I was influenced in a very different way. And I think that that's something that we've often missed in genetic studies. So we were constantly putting up our hands saying, what's going on here? But then we got revenge finally after being rejected for years. We made the cover last month of Nature Human Behavior in an article we published where we wanted to look in more detail at genetic differences across birth cohorts and country. And this is the study if you want to look it up. We think that there's probably a lot of the things that we've been looking at have been hidden due to this variation across country and across birth cohort. So I think the punchline from this is a lot of these big genetic findings have combined data from vastly different historical periods, birth cohorts, and country. And this huge difference in, I was showing you that we found that it was 15% genetic heritability from the SNP-related studies, the Gremel studies, and then once you get to the GWAS studies, you find about 1%, and you're thinking, what's going on? And our study engages in several simulations, and we look across 7 different societies and multiple cohorts and show, hey, wait a minute, it might be related to, I know it's stupid for social scientists, but social context and social environment. So what we show is that particularly for the complex phenotypes such as education, age at first birth, number of children ever born, and to some extent BMI, that once you add in, if you just look at genetics, you get a low predictive factor, but once you add in the birth cohort where they come from, the country where they come from, or the 3-way level interaction, you can actually explain a lot more of the outcome. And we argue, and what we show through several simulations, is also that this hidden heritability or this social environmental context is really important. So it's probably not that important for the biological traits such as height, but it becomes increasingly important for these social or complex behavioral traits. And I know for a group of I presented this in Oxford to sociologists, and they're like, so you're telling us that birth cohort and country is important? It seems a little silly, but it's something that hasn't been acknowledged very much within this field. And then I'll just try to conclude here with another example of just, how we're looking at things differently. And we wanted to look at sort of the neighborhood and also individuals living within a low socioeconomic environment. And we wanted to see if living in a poor neighborhood mitigates the genetic predisposition that you might have to have higher education. So if you have a higher score in this polygenic score for education in years, but you live in a low resource environment, what will that genetic propensity be realized? Because we see that, year on and year out and after different generations, that individuals from poor resource neighborhoods consistently don't seem to do as well in education. It's actually an old idea. Many of you probably know about it, but we have new data where we can look at it. Plomin and many others have found that in poor families, a lot of the variance in education and cognitive ability is actually mostly explained by the social environment. And in twin studies and multiple studies, the genetic heritability always seems in the low socioeconomic environments, it seems to be estimated close to 0. So, and as a sociologist, we're constantly theorizing about these things. And we know that, the mechanisms are social isolation. There's often no adult role models or disorganization, lower social control for your homework, lower quality schools, but even, pollution or noise, and all of these things can influence. So those could be some of the mechanisms that could be operating. We used AdHealth, which is also around 10,000 cases, and it's you have to apply for it, but you can get that data quite easily. It's got 5 different waves, starting with children. It's in the US. And we used the Educational Attainment Polygenic Score. And we did a principal component analysis on neighborhood quality by some of these different indicators. We wanted to know if your neighborhood quality, when you were a child, if that would predict your years of education and your college degree when you were age 24 to 32, but also controlling for these genetic propensity to have higher education. So we looked at polygenic score for education, and I won't go through all of these different models, but we controlled for various things. And then what we found was actually just reconfirming what the twin studies and many studies have previously found, that genetics matters, but it seems to be only when you have resources. So this is the polygenic score, and this is our linear prediction. These are the lower socioeconomic kids that come from lower socioeconomic neighborhoods, and these are the ones that come from higher socioeconomic neighborhoods, and this shows as their genetic propensity to have higher years of education, which has been correlated with cognitive ability. We see that they seem to get more returns from having this genetic propensity to go to school or have more education. So we see these differences. And, there's lots of reasons why, but one of them that we were able to look at in the data set was it could be the educational aspiration of the parents. So indeed, if it was asked how disappointed would you be if your son or daughter did not graduate from college, it definitely, these are again the high socioeconomic group, and it shows that the parents' expectation for them to go to college, it's fairly flat for those that come from low socioeconomic backgrounds, even though they have a much higher genetic propensity and it's correlated with cognitive ability. But these ones, the higher socioeconomic children, are able to meet their genetic potential. And there's lots of different explanations you could think about for that. And I guess just to conclude, the elephant in the room that's been addressed for several times, so a study we're working on in more detail. I'd just like to clarify that all of these genetic discoveries, or most of them, in these 3,000 studies that I mentioned, it used to be about 96% were people of European ancestry, so white people. And now in 2016, they made the huge jump to have 81% of the studies. So most of the studies are in Asian samples. Very few are in African samples. So I guess as a sociologist, it's just important to just think about, since most of these studies are related to, drug development and other factors, it's striking when you see that. And we're looking at this in more detail as well, too. So just to conclude, can we ignore this stuff? I think we can ignore some of the early studies that haven't been replicated. I think we have to be careful with thinking that it's the answer. So as James said, it's not, predetermined genetic determinism. We'll probably only get up to maybe 5 or 6%. Educational attainment is predicting maybe around 11% of the variance of your models. It's pretty high, but it will never take over our social science predictors, and it's always in interaction with the environment. I think it's really interesting when you start thinking about the different theories. So in sociology and demography, the way to look at fertility is often the theory of planned behavior. So it's all about choice and agency and structure, and where do genes play a role in this? So that's something I think that's very interesting, to rethink some of our micro-level theories. And I think it's just could be very interesting to think of new causal pathways in different models that we can run. So what happens if we ignore it? This guy, and people, I'm trying to find the most devious looking geneticist I could, but, there'll be people working on social class and socioeconomic status and occupation, and they'll be defining them, and I think we're masters of measurement. So I think it's really important for us to come in and measure things correctly, because I have talked with people and they said, oh, social environment doesn't matter. And I'm like, well, how did you measure it? Well, we combine income, occupation, and education into one variable, and it didn't do anything. And you think, oh, yeah. So, so I think it's really important because we're very good at measurement, and that's what's lacking. I think also we should think about some of the policy and ethical issues that, it's something I think that's important, and I won't go into detail, but there's been some really interesting studies by Jason Fletcher that's looked at some of the policy implications of looking at this data. And I doubt it's going to overturn our existing findings and what we know, so we don't need to feel threatened, but I think it's useful and it can complement what we know. So thank you very much.
Thank you very much, Melinda.
Okay.
We have time for questions. Again, for those of you online, if you want to tweet your questions, that would be great. Question for Melinda.
Thank you. Just a quick question about statistical methods that you've used throughout all the studies that you've been involved in. So thinking of methods such as regression and sequence analysis, event history, which I think you published on and whatnot. To what extent, I can see how it's very challenging to bring both social and biological data together into one statistical framework. To what extent do you find that commonly used methods that would not are fine and adequate, or do you find yourself challenged by having to amend and adapt existing methods, or perhaps look at more obscure ones that previously didn't be used very much?
Could you repeat?
I'll repeat the question. So you asked about statistical methods and some of the ones that we use, and if we could use commonly used methods, or we had to think of new ways. Well, that's been one of the biggest problems and one of the largest realizations. So I wrote a event history analysis in our textbook. So I was used to using these kind of models, and I thought, okay, they're good. But when we told the analysts that we wanted to look at right censoring, so people that didn't experience the event or a more complex event history model, they said, no, that's too complicated. We just look at it as a linear OLS regression. So we encountered some of those things, that some of the models that we've been looking at to look at our social science phenotypes are not common at all in the GWAS studies. So they often look at OLS regression. In our last study, we even had some issues with people using a logit model, where we had to go into that. So that's been really interesting and empowering, of course, as a social scientist. But for the paper I showed you that was published in Nature Human Behavior, there wasn't so GCTA and Greenwald they weren't able to deal with it. There was no package that could deal with that, so we had to do these multiple matrices and estimate them. They've now integrated it into their package, maybe as a result of our paper, but the things that we're thinking of and doing, there's often not a package or something available. And this data is, like the UK Biobank data is 7 terabytes. So it's not that you just run something quickly. I mean, it's massive, so we're now at advertisement quickly, sorry. We're writing a textbook on how to do this and doing it in a very accessible way. And it's just trying to get over those hurdles if you do need to use a clustered computer, or if you do need to do some of those things. And DNA, there's an open project, and they've donated 9,000 cases that we can use for our textbook, so people will really be able to do this. We'll make it all online and open source, so we're hoping that that will be, and that's NCRM funded us to do that, so that's, you know, one of the things that we hope to give back as well.
And I learned a lot there. That's really interesting. And I'm not familiar with this literature on polygenic risk score for education. But what are the biological mechanisms meant to be there? Are they cognition or conscientious behavior?
Yeah.
What's the story?
So you asked about the polygenic scores for education and what the biological mechanisms were related to biology, cognition, or personality. It's really interesting because as they get more and more findings, it gets more complex, but it's definitely related to cognition. They were relating it to Alzheimer's, cognitive decline, and then forgive me because I can't remember, but there's certain parts of the brain that they were relating it to. And but they do have, as with our fertility study, it is related to certain personality traits as well, openness to experience and different things like that. They have a very detailed, so these papers are short in nature, but then they have 150 pages of supplementary material where it describes in great detail all of these sort of relationships. Because it's written by sociologists and economists, my experience is that you can understand quite a bit of it as well, too, which is interesting. And now, you know, we're replicating it, as are they, and we've got access to 52 different tissue types as well, so it gets really interesting. Yeah.
My question is about this replication, and you have to forgive me because I don't know a lot about the methodologies involved and doing GWAS and getting on polygenic scores. But you talked in some cases about people sort of beating you to it in terms of publishing things. So people actually taking pre-existing analyses done on the tissues and then combining them with other studies, sort of like meta studies. Is this how these studies are sort of built on one another?
Yeah.
Or when people are doing their own studies, they able to get hold of the tissue and then running the actual genetic analyses themselves? I'm not quite clear how it all operates.
So there's, and this is a good thing to tell social scientists. So when we are at a conference and you mention an idea to somebody, the person says, oh, interesting, and usually they don't run back to their office and write the paper. That's been a different experience I've had in this field. So we sign, this is being broadcast, isn't it? Okay, we sign collaboration agreements, but not everyone, that they won't mount a competing study next to us, but not everybody follows that agreement. So I'm Canadian, and I'm from a farm. I was assuming when you signed something that was what you did, but that's not the case. And then another thing is that we make everything open access. So the day we published our paper, we made our polygenic score open access. That's the nature, you know, and that's their requirement. Some of the studies are finding it difficult to find the time to put them on for about a year or two. So that's a difference in behavior. But we were hoping if we put it publicly that everybody could use it. And it's also for exactly the case you're saying. Somebody might have a particular tissue or something, and we want them to do it. That's not our expertise, so we wanted people to use our studies. So, you know, they then jump us on a few things. But in a way, I think, I believe quite a bit in open science, so I think it should be out there to be used and be transparent. Yeah. Yeah, so there's a big discussion in genetics about, so you used to have these, and I showed you some of the results, maybe in the wrong order, but you know, you would have these results from the twin studies, behavioral genetics, saying that something was 50% heritable. So that was the case with age at first birth. If you look at all the twin studies, you would think that it's you know, about 50% related to genetic predisposition, which does seem kind of high, actually. So there could be other factors there. And then we got, a few years ago, this GCTA, or these GREML methods, where you can compare, where you can use whole genome methods, and you can compare genetic relatedness of people. So you don't necessarily need to use the twin studies anymore. You can use GCTA, and that's called SNP heritability, so single nucleotide polymorphism heritability. And then you have the GWAS studies that actually found the genes, and they were predicting around 1% or 2% or 3%. So you went from 50% twin heritability to around 15% SNP heritability, and then to around 1% GWAS heritability. The difference between the SNP heritability, 15%, and the GWAS heritability is what we call hidden heritability. I was coming to the answer. So we wanted to understand, because the twin models we're not so sure. We're thinking they're picking up different things. But with whole genome data, using the same data, you're wondering, why are we finding different things? So that's what we thought, well, maybe it's because you're combining all of these very heterogeneous populations. And indeed, if you just use the UK Biobank that now has 500,000, they're getting many more hits. And the findings are becoming more similar. And that's a homogeneous one country population. So I hope I described it a bit, but we're trying to understand, you know, how is it possible using whole genome data that you have this disparate hidden heritability or differences in heritability? Is it the statistical technique, which everybody has been looking at, or we alternatively said, well, is it differences across, you know, between when my mom had a kid and I did, and different cross-national differences?
So just thought, why do you call that hidden heritability rather than the opposite, which is an overestimation of heritability because you're homogenizing content?
Right. So so if, and I can refer you back to the article. So we have because we went back and forth on this, we have a whole box where we define all of these different kinds of heritability. So there's missing heritability and hidden heritability and all of these different things. And where we found we could probably have the gains would be between the SNP heritability and the GWAS heritability. So, and that's due to this heterogeneity across these different populations and birth cohorts. So, you know, we went through all of these different assumptions, and I won't go into detail, but that's why we focused on that, because that's where we thought the most gains would be.
I might have misunderstood, but I thought it was different between the example there, 1% and 15%. As a consequence of the 1% being a much more heterogeneous sample.
No, the 15%, we had seven populations when we produced that one, and then the 1% had more populations, but it still produced, they both had diverse populations. They were just using different techniques, so something's not making sense. Yeah, so, and it could be related to genetics or the statistical technique, but we're offering a social science alternative.
Could I ask a question about you? You gave at the start some of the dodgy findings coming out from sort of 10 years ago in AJS. What have we learned to prevent that kind of, was that because of the lone wolf approach? I mentioned to some genetic epidemiologists that social scientists should become more involved in biosocial research, and they said no, because look at that, they cited the AJS papers. That's a terrible example of how social sciences use genetics. In fact, I was immediately chastened.
Yeah.
But I was thinking, you know, well, I'm sure if I think about it, there might be really terrible examples of how, you know, genetic epidemiologists might have used social science data.
Exactly.
But I just wondering if if there are things that we've learned from, say, those approaches that were used 10 years ago.
So the question is, should we learn something from the dodgy findings of the last 10 years? Is it related to being a lone wolf? And does it happen on both sides? So the dodgy findings are definitely on both sides. And I think this is some of the things that we're trying to uncover a little bit, like let's look at sex differences or country or cohort. It's not very welcome, I can guarantee you that. You know, we're just focusing on different populations. It seems to be we'll have a study coming out where we show of the 3,000 GWAS, you know, what country they're based in, and it's very revealing. You'll see it. So, and that influences the traits that you look at. And I mean, what Gillian was looking at was very much related to nutrition and related to, you know, development, which is less often the case in these wealthy societies. So, you know, we were talking about that. So I think it influences things, but it's definitely, I would definitely give the advice that if anyone wants to enter in, and I think we're all examples of it, is, you know, you leave the I've learned to leave the biology to the biologists, and I will never get to their level of expertise. I mean, you've seen us, you know, we're presenting it, but it's better that way. And I mean, I was talking with one of our co-authors, and I said, oh, what are we going to do? And you know, to follow up with the study, and he, you know, he let me talk, and then he said, but Melinda, that was what we were discussing, that was 3 months ago. The field has evolved, you know, and we're doing new things, and there's more data, and I think we can't keep up with that, nor should we think we can, you know, because you always need one foot in a discipline, I think, you know, yeah.
On that note, I'd like to thank Melinda and actually all our speakers today. So thank you very much.
Okay, thank you.
And thank you everybody online. The slides will be made available, and the talks will be made available as well. Thank you.